Welcome to another lecture. This lecture is about plant defense mechanisms using secondary metabolites. So to start us off, let's look at this picture here from your book. So it shows a plant and it says virtually every part of the plant is adapted to coexist with the organism in its environment. So you see that the roots are interacting with symbionts and with soil microbes and root pathogens and nematodes and root herbivores, and everything is interacting with the root. And then above ground, there are herbivores nibbling on the leaves, and there are pollinators that the plant wants to attract. There are leaf pathogens that are basically also eating away on the leaf, and other insects that the plant interacts with. And then, of course, there's competition between plants as well. So this kind of interaction is oftentimes done by using secondary metabolites. And so what we have discussed so far in this class in several cases is the secondary metabolites and the production of these secondary metabolites. We talked about phenolics, terpenoids, alkaloids, and so forth. We looked at the interface between primary and secondary metabolism. So where do the compounds, the precursor materials for the secondary metabolites come from? And today what we're going to talk about is how these secondary metabolites are used in interactions with the external environment, mostly with respect to biotic stress responses. Okay, so what I want to talk about today a little bit is how plants use secondary metabolites to defend themselves against attack from the outside. And there are four major categories that you can classify these responses. One of them is called the constitutive defenses. Then there's the induced defenses. There are, sort of generally speaking, defenses against pathogens, and this involves a lot of pathways that we will discuss. And then there is herbivore-induced volatiles. So these are compounds that are volatile, and we'll talk about those at the end. So the constitutive defenses are the ones that are always present, constitutive meaning always there. And many of the examples are those that we have already discussed in the previous lecture about secondary metabolites. So those are substances that a plant will make and will store in its cells, and they're just always there. So it's something that doesn't have to be induced at the time of attack. It's just something that the plant makes and stores and has ready in case something starts nibbling at the leaf. So we won't talk about much about these today. We're going to start here with number 2 and then move on to 3 and 4. So the main induced defense pathway is one that uses a plant hormone that's called jasmonic acid. So a hormone in general is a small organic substance. Sometimes it's a peptide, and sometimes it's an organic substance that by definition is made in one cell and then delivered to a different cell where it acts. And that's sort of also the definition for animal hormones. It's made in a gland or in a certain compartment, and then it's transported elsewhere. With plants and plant hormones, that definition had to be augmented a little bit, because these compounds that act as hormones can oftentimes also act exactly in the place where they were made, because plants don't really have glands like animals do, where they produce these hormones. But certain cells are specific for producing certain hormones. And we'll talk about more hormones later on as well. But today, the hormone that we'll focus on is jasmonic acid, and that's the one that is induced, that creates these induced defenses. So just a few general things about jasmonic acid. So it serves as an activator of defense protein transcription. It's not exactly know what turns on jasmonic production. So jasmonic acid can be there, but it can also be induced in its production. It is increased after herbivore damage. So when animals start nibbling on the plant, it has been shown that there's more jasmonic acid. And we'll talk about how jasmonic acid then leads to the defense responses but not so much what turns on the production of jasmonic acid itself, with the exception of one compound that is known to turn on jasmonic acid. I'll show you that in the next slide. And then just as a definition, we'll talk about elicitors. Elicitors are just something, they elicit a response, right? They turn on a response. These are specific compounds, oftentimes found in organisms that are attacking the plant. So they could be, for example, in insect saliva, or they could come from bacteria, and they elicit a specific plant response. So I'll talk about these elicitors. Okay, so let's go back to jasmonic acid. So we're in the middle of talking about induced defense initiation via jasmonic acid. So let's first see how jasmonic acid is made. So when there is some sort of attack, either through these herbivore elicitors or through a compound, that's the one that I had mentioned earlier. Well, I hadn't mentioned the name yet. I just said there is one other compound that turns on jasmonic acid production, and that is called cysteamine. That is a little plant protein that is also induced by herbivory. And then, that is not really known what exactly turns on cysteamine. So what is known is that cysteamine and jasmonic acid are turned on by herbivory. And then the downstream responses of that is quite well understood, and that's what we'll focus on today. So here's our first step in the jasmonic acid defense response. Herbivory turns on jasmonic acid and cysteamine production. Okay, so how is jasmonic acid made? Well, it is made in the chloroplast with the help of the peroxisome and with the help of several pathways that also are used for other pathways in the plant, specifically beta oxidation. So we talked about beta oxidation a little bit. Beta oxidation is used for the breakdown of fatty acids. And as you can see here, this starts with linolenic acid, which is a fatty acid, polyunsaturated fatty acid. And through a number of steps, it sort of turns over and produces this circle, cyclization, and then is transported into the peroxisome, where the beta oxidation steps occur. So basically chopping away this end that looks like a fatty acid tail, leaving jasmonic acid. So this is jasmonic acid here. And the jasmonic acid is then exported into the cytosol. So those are the first two steps in the responses using jasmonic acid against attack. So what does the jasmonic acid then do cellularly in order to transmit the invasion or the attack, the perceived attack? So for that, I have this pathway, this transcriptional activation pathway that I want to discuss with you. So the next step is that jasmonic acid here, this is this green little diamond, binds to an amino acid that's called isoleucine. So when it binds to isoleucine, that activates jasmonic acid. So now jasmonic acid is able to bind to a protein complex. That protein complex is called the SCF-COI1 complex. And you see that it's made up of two different proteins. So I say here jasmonic acid binds to isoleucine and then to the SCF-COI protein complex. This way of writing the two gene names or protein names in this case indicates that they're complexed with each other. And then these four components together, they can then bind to the JAS protein bound to the MYC protein. Okay, one step at a time. So what are the JAS and the MYC protein doing here? Well, they act as transcriptional repressors. So the MYC sits on a region of the gene that is inhibited, so the promoter region of the gene. As it's sitting there, bound to JAS, to the JAS protein, the transcription is inhibited. And it's sitting on promoters that are jasmonic acid response genes. So these genes are genes that could be turned on in the case of the need for defense. So in the case of attack, these genes are required. But as long as there's no attack, the genes are turned off, actively turned off by the binding of the JAS protein to the MYC protein to this promoter region. So now when there is an attack and jasmonic acid is made, and this jasmonic acid binds to isoleucine and then binds to the SCF-CoI complex, these together are also known as members of a large family of F-box proteins. So when these bind together, they can then bind to the JAS protein. And F-box proteins in general, and this one as well, have the ability to degrade their targets. So the way that they degrade their targets is by binding to them and then adding what's called a polyubiquitin tail to the target. So polyubiquitin is a small a peptide. And when it's bound to anything, it's sort of sometimes referred to as the kiss of death. What then happens when polyubiquitin is added to a protein is that that entire protein is mobilized and moved to the proteasome, to the 26S proteasome, which degrades proteins. So in it goes into this protein, and then out come little bits of amino acids that are spit out of the proteasome. So just to recap, this binds to this. This will then act as an F-box protein that polyubiquitinates its targets, and the target is specifically made clear to this complex by the binding of jasmonic acid and isoleucine to the target. There it goes. MYC stays. And MYC can now act as a transcription activator. So now it activates these genes that it previously, while bound to JAS, was inhibiting. So this inhibition of this gene here is just one example. I'm writing here, one of many response genes is JAS. So the genes that are inhibited, as long as there's no attack, are a number of different proteins that can act as helping the defense of the plant. So for example, this purple gene here could be an example, or could stand for an example, such as a protease. So a protease, when that is made, will inhibit proteins from the attacker, for example. Amylase inhibitors. So that could be, amylases could be in the saliva that are trying to digest and degrade starches in the plant. So if you have an amylase inhibitor, then the attacking insect is no longer able to break down the sugars. Lectins are also examples of defense proteins that are made here, indicated by this purple gene or this purple line. And those are proteins that bind to carbohydrates and make them indigestible for the herbivores. So these lectins will then bind to the plant carbohydrates and make them unusable for a caterpillar, for example, that is chomping away at the leaf. Okay, so here I say it also binds and makes this gene, one of these genes that are transcribed is also the JAS protein itself. So why would that be? That's the one that turns everything off, right? So that gives a negative feedback response, and that is so that only as long as jasmonic acid is around, only as long as this defense is induced by an active attack, will the transcription of these genes be going on. So as soon as there's more JAS protein that turns its own production off, if there's more of that and less of this, so less of the jasmonic acid, then it basically turns itself off again. And that's good because it means that these relatively expensive proteins are only made during the time when they're absolutely needed and not made when the plant is not under attack. So we have a negative feedback loop. We have JAS and MYC turning off transcription of a whole slew of genes. There's just one shown here as an example. They're all called jasmonic acid response genes. And when they are turned on during an attack, then they make these protease inhibitors, the amylase inhibitors, and these lectins. All right, so that is on the cellular way, on the transcriptional way level, how this is regulated. So let's take a step back here and look again to see where this whole defense mechanism occurs. So we have a cell here, right? So I talked about the peroxisome, the chloroplast, how jasmonic acid is made, and then this response that I just showed you in the previous slide. So all of this happens what's called on the local level. So that's exactly where the caterpillar is. That's where these responses occur. Now, there are ways for the plant to make this systemic, so to move a response from one place that is currently being under attack to a place that is not yet under attack, but where chances are that the caterpillar will move to relatively soon. And so there's two ways. The first one I had already mentioned, that's the systamen. So systamen is a protein that will turn on the production of jasmonic acid, and it, again, by itself is activated by attack. So this happens on the local level. So systamen is a protein that needs to be activated by cleavage. So the cleavage of the protein from prosystamen will release systamen, which can then act on neighboring cells, and these neighboring cells will then turn on the production of jasmonic acid and turn on this pathway. But jasmonic acid itself can also move away from the local level to the systemic level through translocation in the phloem. So it can go into the vascular tissue and then can be then moved to other target cells, potential target cells for future attacks. So that's where this local and the systemic part comes into play. So the plant can move this from one place where it's currently being under attack to another place where attack is maybe imminent. Okay, so that is jasmonic acid and how it's working as a defense mechanism. Now let's look at some defense mechanisms that are specifically evolved against bacteria, viruses, and fungi. So again, we have a number of categories, and I want to walk you through some categories, and I want to illustrate those with some examples. In this first case, it's all about detecting certain elicitors from pathogens, from bacteria, viruses, or fungi. And so what happens here is that the plants have evolved over time an ability to recognize these pathogen-derived substances. So here's a bacterium, it has a flagellum, and just by being who it is, it makes certain compounds, and these compounds are potentially recognized by the plant. So the plant has evolved receptors against certain elicitors, against certain compounds that are made in this bacterium, in this particular example bacterium here. And these are categorized into two different categories. One of them is called the microbe-associated molecular patterns, or MAMPs. And they incorporate the recognition of whole classes of compounds. So for example, when I say whole classes of compounds, bacterial flagellin, that is that's the protein that is associated with the flagellum. There's lots of different types of bacteria. They all have flagella, and they all have this bacterial compound, flagellin. And so all of them have that just because they have the flagellum. And so that's a whole class. That's what I mean with a whole class. So all of these, this whole class of bacteria that have flagella will also have flagellin, and flagellin can be recognized by plants as a substance that is associated with bacteria. And so that can then act as an elicitor, and these elicitors bind to certain receptors, and they will start downstream responses. We'll talk about these downstream responses in a second. So those are these MAMPs. Now there's also something that's called a gene for gene resistance, and that's something like if you were to study plant pathology intensively, maybe in grad school or so, you would hear a lot about these gene for gene resistance pathways that have evolved in a co-evolutionary dance, if you will, between the plant, a specific plant, maybe even a specific species of plant, and a specific a pathogen that has specialized on a specific species. And so while the bacteria might have specialized on a specific plant species to infect, the specific plant species might have co-evolved a specific receptor for a specific substance. And that's why they're called gene-for-gene resistant. So one gene in the bacterium has made a certain substance that it might need for invading the plant, but the plant then has evolved a gene that gives it resistance against the gene and the product of this gene from the pathogen. And then again, the pathogen might have evolved something that will make it able to withstand this gene resistance. So it's a back and forth, back and forth. I have an example of that at the very end. So gene-for-gene resistance are co-evolutionary learned resistances. The one thing that I always got hung up on is this idea of the elicitor. The term means that it elicits something. It's made to elicit a response. That's not really the case here in terms of the reason for why the pathogen makes this elicitor. The pathogen has no intent on eliciting this response. It just happens to make these compounds just because that's who it is, right? It has a flagella, so it makes has a flagellum, so it makes this particular substance that will elicit the response, or it has other properties that the plant will then recognize as a pathogen. So don't think about the elicitor as something that is intentionally produced by the pathogen. It's something that sort of, unintentionally is produced by the pathogen and then used by the plant in its own defense. Okay, so we talked about these, the gene-for-gene responses and the elicitor detection, but there are also pathways in the plant that are called the hypersensitive response. So the hypersensitive response is a different type of response to bacteria than the ones that I talked to you just about. And under the term of hypersensitive response, a number of signaling pathways are meant. So what happens in a hypersensitive response is that oftentimes you get a burst of reactive oxygen species. So for example, H2O2, that's the main reactive oxygen species that is made. So the plant recognizes that it's being infected or invaded by bacteria, certain viruses or fungi. It produces this H2O2, which will oxidize invading cells. It's basically chemical attack. It will try to oxidize these plant cells, these invading cells. But as it's doing that, H2O2, you already know, is also not good for the plant itself. So it, in that process, kills plant tissue as well. So it kills some of its own plant tissue. That has two effects. One, it sets in motion downstream responses, and I'll talk about those in a second. But it also helps by sort of leaving, it's just like scorched earth, right? The scorched earth tactic, where you leave nothing for the invader that the invader can use. And so the plant will burn its own tissue. It will basically kill its own tissue so that that cannot be used by the invading bacterium. And maybe you have to sacrifice a leaf, but at least you don't have to sacrifice another leaf or the rest of the plant. So that is what happens with this burst of H2O2. But H2O2, as well as other signaling molecules, nitric oxide, for example, and calcium, can also be produced and act as a second messenger. So this H2O2, when it has this reactive oxygen burst, will try to kill invading bacteria as long as they're still on the outside or just coming into the leaf. But at the same time, it can also, together with calcium as a second messenger and nitric oxide as a second messenger, can turn on these responses. So this certain set of responses that then downstream will help the plant to fend off these invading bacteria if the initial response of the reactive oxygen species hasn't helped or hasn't killed all of the bacteria. So what these signaling compounds do is they produce a number of secondary metabolites. So for example, phytoalexins. Phytoalexins are just a general term for attack-induced antimicrobial secondary metabolites. And so you see that they can be turned on by this set of signaling molecules in addition to salicylic acid. Lignin biosynthesis, so lignin is this woody substance, we talked about that, it's a phenolic, so that can be increased in its production so that that helps the plant to seal itself off on the outside against invasion. All right. Another category of defenses against pathogens are systemic acquired resistance. So that's a term that involves a number of volatile compounds. Now, those are not the volatile compounds I put in category 4. These are substances that are specifically induced after attack by bacteria. For example, here I show you salicylic acid as one of the compounds that's being produced in these initial response steps. Here's salicylic acid. Here's the pathogen infection. So that is a compound that is initially induced, but systemically, so it doesn't act systemically just yet right after the infection, but just like the jasmonic acid that I showed you at the beginning, when I showed you that it can act locally but then also become systemic, the same is true for salicylic acid. So it can act locally, but then it can be transported through the plant, and then it can be volatilized. So by putting an ester on it, right, this is the methyl ester of salicylic acid called methyl salicylate. That can then be volatilized out of the plant and can be perceived through receptors in a neighboring plant. So that can in fact increase resistance of a plant that has not even been attacked yet. Okay, so that then leads to systemic acquired resistance. So that is the term that will describe responses in parts of the plant or other plants that haven't been attacked, but that are still producing these secondary compounds as defense molecules in case of attack. Okay, let's put this all together. I showed you little snippets of this before. I put this in 3 categories, but really there are all these pathways that are intertwined with each other. And so this is trying to show you how they are intertwined with each other. And I put these numbers on there. Those are the numbers that go with these individual steps that I had here. Number 1, number 2, and then number 3, the systemic acquired resistance. So how are all these different steps put together in one pathway? Well, you see here the elicitor, you see the receptor, you see the signaling compounds, and they can come through elicitation, or they can come through calcium influx by recognizing a compound, a fungus or a bacterium from the outside. They can come through H2O2, which then again came from the reactive oxygen burst, which was found. Which was turned on by the receptor through this elicitor. So multiple pathways, many layers of ways for the plant to come up with all of these types of responses on the local level, and then can also move it to the systemic level. Okay, so I wanted to show you one example where this co-evolutionary pathway between plants specifically evolving to produce receptors that recognize specific elicitors can also lead to the production of certain compounds in the plant, in the bacterium, that then take advantage of certain plant pathways. Let me show you what I mean. So here's an example of what I call a co-evolutionary arms race. So In the example of bacteria, you have salicylic acid that is upregulated, and one of the genes that are regulated through salicylic acid is called NPR1. And NPR1 specifically helps the plant to defend against bacteria. So it has a bunch of downstream responses, other genes that are then turning on all these pathways that I generally talked to you about before. When fungi are the source of the infection, oftentimes jasmonic acid is upregulated more so than salicylic acid. And jasmonic acid, via the pathway I told you at the beginning, through this interaction with COI and the transcription factor MYC that sits on these jasmonic acid response genes, there's a whole set of defense genes that are specific against fungi that are upregulated through this pathway. Well, in order to, I guess you could say, in order for the plant to specialize on one pathway versus the other pathway, so that it doesn't produce a bunch of secondary compounds or turns on response genes that it may not necessarily need when it's infected by a bacterium and not by a fungus, or when it's infected by a fungus but not by a bacterium, there are these interactions that have evolved in the plant where NPR1 not only, this is a protein, right? So when that gene is expressed, that protein turns on defenses against bacteria as a transcription factor, but it also inactivates or represses the jasmonic acid COI pathway. So basically, it helps the plant specialize to have bacterial defense responses when it's infected by a bacterium, and not at the same time also upregulate its fungal defense responses. And vice versa, MYC2 will inhibit NPR1 so that it can concentrate on the fungal response when infected by fungi. So there's a particular type of bacterium that's called Pseudomonas syringae. It's a bacterium. It would require a bacterial defense response. But Pseudomonas has evolved a secondary compound of its own that's called coronatin. Coronatin activates MYC2, which activates defenses against fungi. And MYC2 inactivates NPR1, which activates defenses against bacteria. Now remember, Pseudomonas is a bacterium. So it invades the plant. It turns on the pathways that the plant uses to put a bunch of energy and activity against producing something that will help it fend off something that it's not infected with, and at the same time usurp its own pathways by turning off these pathways against bacteria so that it can take advantage of the fact that the plant is basically involved in mounting a huge response against something that it's not infected with. So that way you have this coevolutionary tit for tat where something gets evolved and then is very effective in allowing this particular pathogen to invade the plant and take advantage of it. Okay, so the last part that I want to talk about are these herbivore-induced volatiles. So in general, herbivore-induced volatiles, we already talked about volatiles that can be used to induce systemic responses in other plants, in neighboring plants. But what is meant here is that volatiles are produced by the plant that act as feeding deterrents or insect repellents, and also to signal to other plants. But when I say here that it acts as feeding deterrents and insect repellents, one other thing that it does is it also attracts herbivore enemies to the plant itself. So it's not just for the benefit of the neighboring plants, it's also for the benefit of itself. Let me show you what I mean. So here's a plant, it's being attacked by this herbivore above ground or below ground, and it's now producing these HIPVs, herbivore-induced plant volatiles. So it's an induced response. So what happens then? Feeding causes the production of volatiles. So these are esters, aldehydes, anything that can evaporate quickly and become can go out into the air, and that is then these volatiles then, first of all, they leave a residue on the leaf, so they make it oily or bitter, I suppose, not very attractive for the attacker. But they can also then attract these natural enemies. This is a little parasitic wasp, and the parasitic wasp then will be basically called for help. The wasp finds its target, which is this caterpillar, and what it likes to do is to put its eggs into a caterpillar, and then when the eggs hatch, the little larvae of the wasp will eat the caterpillar. And so these volatiles call for help, and then these predators, basically my enemy's enemy is my friend, will then help get this invasion of these caterpillars under control. That's one way how herbivore-induced plant volatiles work, but they also act as repellents so that other herbivores don't even show up, and they act as a way to induce responses in neighboring plants. So that must be an evolutionary adaptation where the plant stands to gain, even if itself is lost to the attack. It stands to gain from having conspecifics in the neighborhood produce at least its seeds and get the genes out that way, even if it can't defend itself. But, not all is lost yet. It's still trying to defend itself, but it's also trying to alert the neighbors. So those are the three categories I wanted to talk to you about today. You see that there's a lot of interaction. You can't really, neatly put them into boxes and into different categories. There's a lot of overlap in ways of putting these responses together into a way for the plant to defend itself against attacks. But one of the main points that I wanted to make, and that I was hoping you will see in this from this lecture, is that many of these secondary compounds that we discussed at the beginning of last lecture, where we looked at how they are made, well, here you see an example of how they are put to good use for the plant. And that's all for today. I'll see you in class.